Non-tuberculous mycobacteria, or NTM-related lung disease, has become increasingly prevalent over the last 2 to 3 decades. Although there are approximately 190 known species of NTM, only a fraction of them are associated with lung disease in humans. These organisms are found worldwide. However, accurate perspectives of NTM epidemiology can be challenging because NTM infections are not typically considered reportable. Common species worldwide include the Mycobacterium avium complex, or MAC, and Mycobacterium abscessus. MAC are slow-growing mycobacteria that encompass 12 mycobacterial species. The 3 most common are Mycobacterium avium, Mycobacterium intracellulare, and Mycobacterium chimera. In comparison, NTM isolates that can be cultured in less than 7 days are considered rapidly growing mycobacteria, or RGM. They comprise species including Mycobacterium colonae, Mycobacterium abscessus, and Mycobacterium fortuitum. MAC and Mycobacterium abscessus are particularly prevalent in the United States, with MAC identified in more than 80% of NTM cases. These organisms can be found in environmental sources that include drinking and natural water, water aerosols, such as are encountered with indoor hot tubs, soil, and dust, all of which can be ingested or inhaled. Exposure to NTM does not result in infection in most people. However, evidence indicates that infections that do occur are transmitted from the environment and not person to person. 2 main theories of overall susceptibility include airway-focused issues, such as impaired defenses or ability to clear secretions, and underlying immune deficiencies. NTM lung disease presents in 2 main clinical manifestations. The nodular bronchiectatic form results in airway scarring, mucus clearing impairment, and development of nodules and consolidation, particularly in the right middle lobe or lingular lung tissue. The cavitary form of NTM lung disease tends to affect the upper lobes of the lungs. If left untreated, progressive cavitation and fibrosis can occur, resulting in respiratory failure. While treatment may not be indicated in every case, resistance to NTM-directed therapy exists. Mechanisms of resistance are classified as intrinsic, adaptive, or acquired. Intrinsic resistance highlights the natural resistance to drugs, which is possible through the NTM structure. A thick hydrophobic double membrane cell envelope creates a barrier that reduces drug permeability. Modifications in the cell wall layer, called morphotypic antibiotic resistance, also produce varying degrees of resistance due to changes in cell wall permeability. Adaptive resistance occurs in specific environments. For example, some NTM species will induce gene expression when drug exposure occurs, resulting in reduced binding or modification of drug target binding sites, rendering the drug inactive. Finally, acquired resistance can occur through genomic mutations inherited through NTM replication. Prolonged and inadequate treatment regimens have contributed to mutation development in target and related genes, resulting in high-level resistance.